Hello, my name is Joshua Gabi. I welcome you to Coordination and Control. Questions 31 to 35. Question 31. Explain the events that occur during saltatory transmission conduction. B. Roman number 1. With the aid of a systematic diagram, outline the basic organization of vertebrate nervous system. B. Roman number 2. Distinguish between somatic nervous system and the peripheral nervous system. Part C. Describe the physiology behind drug addiction in people who overuse drugs such as cannabinoids and marijuana. Part A. Explain the events that occur during saltatory transmission or conduction. Saltatory conduction in a myelinated axon, action potential are only produced at the node of Ravian in a myelinated axon. One node depolarizes the next node so that the action potential can skip between the nodes. As a result, saltatory, or what we call leaping conduction in the myelinated axon, is more rapid than conduction in unmyelinated axon. So we are looking at the saltatory conduction, and when we talk of the saltatory conduction, it is a leaping conduction. We have the axon. The axon is this middle part of the nerve. The oxoplasm makes up the axon. Then outside, there are membranes that cover the axon. Those membranes, we call them the myelin sheath. But when they are covering, they leave a gap, and then another one starts up, and then leave a gap, and then they cover up. The places that are left, those are the ones that we call the node of Ravian. Node of Ravian. So those are part of the axon which are not covered by the myelin sheath. So what happens is that when the action potential is produced at the node here, when the action potential is produced at the node, that means that the inside the axon becomes more positive than the outside. Then we say we have the action potential. Now this action potential, when it is formed, it will now cause the action potential to be taken to the next node, or it will depolarize the next node. So what is going to happen is that the action potential is just going to be skipping from one node of Ravian to another node of Ravian. And as it keeps on skipping like that, then there is going to be a rapid conduction of the impulse through the axon, because the impulse is not just passing within the axon, but it is just skipping from one node of Ravian to another node of Ravian. But this one happens only in the myelinated axon. So the speed is faster in the myelinated axon than the conduction in unmyelinated axon. Unmyelinated axon, those are the ones that do not have the myelin sheath. Part B. With the aid of a schematic diagram, outline the basic organization of the vertebrate nervous system. Now we have what we call the central nervous system. The central nervous system is divided into two. We have, the nervous system is divided into two. There's what we call the central nervous system, and another one is called the peripheral nervous system. Now, the central nervous system is made up of the brain and then also the spinal cord. That's what makes up the central nervous system. Now, another part of the nervous system is the peripheral nervous system, and the peripheral nervous system has go to what we call the sensory pathway, and then it has also the motor pathway. Now, when you come to the motor pathway is divided into two. There is the voluntary nervous system, and we have also the autonomic nervous system. The voluntary is the same as the somatic voluntary nervous system, and the voluntary nervous system is the one now that we use under conscious, under control. It is the voluntary nervous system. But the autonomic nervous system is the one that is having no control. We don't have any control. It is autonomic. It is part of the motor pathway. Now we have, under the autonomic nervous system, we have the parasympathetic division and then the sympathetic division. That is the basic organization of the vertebrate nervous system. Part B, Roman number 2. Distinguish between somatic nervous system and the peripheral nervous system. The somatic nervous system is a component of the peripheral nervous system, which controls activities where an individual has conscious control over. And it includes the sensory neurons that sense the external stimulus, and the motor neuron which supply the skeletal muscle. So that is the peripheral nervous system. Our peripheral nervous system, this one is the one that consists of the sensory pathway, or what we call the sensory neurons, the one that sense the external stimuli and send them to the central nervous system. Then again it has the motor neuron or the motor pathway. Now the motor pathway is the one now that sends the response to the neurons like the skeleton and so on. So in the motor neuron or in the motor pathway, that's where we have the voluntary or the somatic nervous system, and then we have the autonomic nervous system, which is divided into the sympathetic and then the parasympathetic nerve division. The autonomic nervous system, on the other hand, is part of the peripheral nervous system that controls activities where an organism has little or no conscious control. Part C. Describe the physiology behind drug addiction in people who overuse drugs such as cannabinoids like marijuana. In a normal synapse, the neurotransmitter binds to a transporter molecule and is rapidly reabsorbed after it has acted. When a drug molecule binds to the transporter, reabsorption of the neurotransmitter is blocked, and the postsynaptic cell is overstimulated by the increased amount of the neurotransmitter left in the synapse. The central nervous system adjusts to the increased firing by producing fewer receptors in the postsynaptic membrane. The result is addiction. And when the drug is removed, normal absorption of the neurotransmitter resumes, and a decreased number of receptors create less sensitive nerve pathway. Physiologically, the only way a person can then maintain the normal functioning is to continue to take the drug. Only if the drug is removed permanently will the nervous system eventually adjust again and restore the original number of receptors. Number 32. Describe the structure of a synapse. Part B. With the aid of appropriate graph, explain the membrane potential changes that occur in a neuron right from rest to transmission of a successful impulse. C. Explain the physiology of acetylcholine as a neurotransmitter substance. Part A. Describe the structure of the synapse. Let's first look at our synapse here before we go. We have the axon. This is the neuron bringing to the synapse, and we have the synaptic vesicles. These are containing the neurotransmitters These ones, the smaller ones, or these ones that we are seeing, they are synaptic vesicles. And then we have the mitochondria. Mitochondrion, we know they are sources of energy. And we have what we call the postsynaptic membrane. The postsynaptic membrane is this membrane at the end of the synapse. That's what we call the postsynaptic membrane. Then we have the neurotransmitter in the synaptic cleft. The synaptic cleft is the space we have between the synapse and the next neuron. And now we have this side, we call it the presynaptic membrane. And then this side, it is going to be called the postsynaptic membrane. The postsynaptic membrane is the whole of this one. It is the postsynaptic membrane of the next neuron. And we can see that when the neurotransmitter is released from the presynaptic membrane, it comes into the synaptic cleft or the synaptic space. Then it will go to the postsynaptic membrane and it will depolarize. It depolarizes the postsynaptic membrane. The neuron whose oxygen transmits the action potential to the synapse is the presynaptic cell. So here, this neuron, which is bringing the action potential to the synapse, is the one that we call the presynaptic cell. While the cell on the other side of the synapse is the postsynaptic cell. So this cell, this side, on the other side, is what now we call the postsynaptic cell. Between which is the synaptic cleft that has a thin film filled with a fluid. So between the presynaptic cell and the postsynaptic cell is a space in between them, and that space here is what we call the synaptic cleft, and it is filled with the fluid. The presynaptic membrane has calcium voltage gated channels. So this presynaptic membrane, it has gates which we call calcium voltage gated channels, which open and allow calcium to enter into the presynaptic membrane or cell from the synaptic cleft. The end of the presynaptic axon is swollen and contain numerous synaptic vesicles, which are each packed with a chemical called the neurotransmitter. So we are looking at the presynaptic axon. This one is the one that we are saying it contains very many synaptic vesicles. All these ones that you are seeing, those are all synaptic vesicles, and they are filled with a chemical called the neurotransmitter. The neurotransmitter diffuses rapidly on the other side of the cleft and binds to a specific receptor protein on the postsynaptic membrane. So when this neurotransmitter in the synaptic membrane comes to the presynaptic membrane, it is released and it will diffuse very fast, and it will come and attach itself on the specific receptor proteins on the postsynaptic membrane. Part B still. With the aid of an appropriate graph, explain the membrane polarization potential changes that occur in the neuron right from rest to transmission of successful impulse. So we have our membrane here. When you look at our membrane here, vertically we have the membrane potential in millivoltage, and then this side we have the milliseconds. Now we have the millivoltage here from negative 100. We are seeing the resting potential. Resting potential is here. It is around negative 60. And then you can see the threshold potential that can initiate an action potential is around here, 60 something. And when it reaches there, the resting potential, now the membrane will become depolarized and we shall form an action potential. Then an action potential, after the impulse has passed, then the membrane will become repolarized. When it repolarizes, it will go back to the resting potential. Now when it goes beyond the resting potential line, then we say it has been hyperpolarized. Hyperpolarization, which takes some few seconds, and then it will go back to polarization space, back to the resting potential. At resting membrane potential, some potassium channels are open and the sodium-potassium pump are active. So when we come to the resting potential here, at the resting potential we are saying some potassium channels are open, meaning that they allow the potassium to get out of the neuron to go outside. And then there is also the sodium-potassium pump. Sodium-potassium pump is also active, pumping out 3 sodium ions and pumping in 2 potassium ions. So when that one continues to go on, then the membrane is going to be polarized, having the inside more negative than the outside. Then we say the membrane is at a resting potential. In respect to the stimulus, the cell begins to depolarize, and once the threshold level is reached, an action potential is produced. So when the impulse or the stimulus is coming, what happens is that now the membrane becomes depolarized, meaning that as the stimulus arrives, sodium ion gates are going to be opened and the sodium ions will rush into the oxygen, making it to become more positive. And when it becomes more positive, we say it is becoming depolarized. Now, when that depolarization reaches the threshold, if the depolarization reaches the threshold level, then we are going to get an action potential. The action potential is going to be fired because we have reached the threshold level. And when the threshold level is reached, an action potential is now produced. Rapid depolarization occurs. The rising portion of the spike. Because sodium channels open, allowing sodium ions to diffuse into the oxygen. At the top of the spike, the sodium channels close and the potassium channels that were previously closed begin to open. So this rapid rise is because the sodium channels have been opened and a lot of them entered into the oxygen, causing the depolarization of the membrane, and therefore the action potential was formed. Now when it reaches the top here, now the sodium channels are going to close, meaning that there is no more allowing sodium ions to enter into the oxygen. But again now, the potassium channels are going to be open. When they are open, now this will allow the potassium ions to get out of the oxygen. So that one is going to cause the decrease in the positive charge inside the oxygen, which is going to lead to what we call repolarization. With the potassium channels open, repolarization occurs because the diffusion of potassium out of the oxygen. An undershoot occurs before the membrane returns to its original resting potential because of the potassium exflux due to delayed closure of the potassium channel and the influx of choline through the open chloride channel. So because the potassium channels are opened, there is going to be repolarization. And when repolarization is taking place, that means that our membrane is, or our oxygen is becoming more negative because a lot of potassium ions are leaving the oxygen, making it to become repolarized. But we are saying that there is going to be an undershoot. There is going to be an undershoot, and that undershoot occurs when it is beyond the resting potential. And our resting potential, when you look at our resting potential, is this one. So when we undershoot, we go below the resting potential. That undergoing below the resting potential is what we call the hyperpolarization. And what brings about the hyperpolarization is the delayed locking of the potassium channel. so that they don't allow more potassium channel to get out. Because the more potassium channels that go out, the more the oxygen becomes negatively charged, and therefore the membrane potential goes more negative and beyond the resting potential. Then we say that the membrane is hyperpolarized. Now, also the chlorine channels, chlorine ions are still flowing into the oxygen because of the chlorine channels which are open, and that one makes the oxygen to become more negative, so it becomes hyperpolarized. Therefore, when time reaches and potassium channels are closed, then the membrane will go back to the normal, which we call the resting potential, because it will rise and then it goes back to the resting potential, and we shall say now it has been repolarized and it has gone back to the resting potential. Part C. Explain the physiology of acetylcholine as a neurotransmitter substance. Acetylcholine crosses the neuromuscular junction or the postsynaptic junction. Acetylcholine binds on the receptor protein in the postsynaptic membrane, and thereby causing the ion channels within those proteins to open. The opening of the chemically regulated channels permits sodium to diffuse into the postsynaptic cell at a faster rate than the potassium that diffuses out. As a result, that site of the postsynaptic membrane produces a depolarized polarization called excitatory postsynaptic potential. The excitatory postsynaptic potential then opens the voltage gate channel of sodium and potassium that are responsible for action potential. In skeletal muscle cell, the action potential stimulates muscle contraction. For cholinergic synapse, the excitatory postsynaptic potentials produced must then travel through the dendrites of the cell body to the initial segment of the axon of the adjoining neuron, where the first action potential will be produced, provided the excitatory postsynaptic potential depolarization is above the threshold needed to trigger the action potentials. The elimination of acetylcholine is achieved by an enzyme in the postsynaptic membrane called acetylcholine stellase, which cleaves acetylcholine into acetyl and CoA and choline, which are its inactive fragments. Number 33. Discuss the functional characteristics of the skeletal muscles. Part B. Explain the concept of echolocation in bats. And Part C. State the structural adaptations of bats to enhance echolocation. Discuss the function characteristics of skeletal muscles. Excitability. The ability to respond to chemical and electrical signals are neurogenic. Contractibility. Ability to shorten and cause movement of the structures to which the muscle is attached. Extensibility. The capacity to stretch to normal resting length after contracting. Elasticity. The ability to return to the original resting length after the muscle has been stretched. Explain the concept of echolocation in bats. Echolocation is the use of sound made by the animal itself to avoid obstacle during flight and locate food in total darkness. Bats, they generate discrete pulses of ultrasound via the larynx and emit the sound through the open mouth or nose. Then determine the time it takes for the sound to return using the method of sonar to locate themselves and other objects in totally dark environment. This is achieved by the characteristics of the echo. By using the information, the bat derives information on distance, direction, velocity, and the nature of the refracting object, and thus makes a decision to avoid an obstacle, to catch its prey, or dip for water. The bat can also as well access more than one target at a time, perhaps perceives the 3D geometry of its surroundings. Part C. State the structural adaptations of bats to enhance echolocation. It has a unique larynx whose muscles are enlarged, the cartilage are fused, and ossified. This makes a rigid framework. The pinners are large and enormous with a tragus. The cochlea is enormously enlarged with the basilar membrane narrow and tightly stretched. Constantly open nose, maintaining a continuous and independent moving back and forth sound. The nose has an epidermal modification like a horn, and this directs the sound forward. Possession of a nose leaf, an epidermal modification that amplifies and reinfuses sound. Distinguish between binocular vision and stereoscopic vision. B. Explain the following phenomena. When a person moves from bright sunlight into a dimly lit room, objects in the room cannot at first be seen, but they gradually become visible. Part 2. The dimly lit room objects are only visible in black and white. Part 3. Some nocturnal animals, like the cats, close their pupil to a vertical slit, squint in bright light. Part 4. When trying to see a faint star in the sky, it is better to look slightly on one side of it rather than directly at it. Part 5. If both eyes are open and you press on the side of one of your eyeballs, you experience a double vision. Part A. Distinguish between binocular vision and stereoscopic vision. Binocular vision is vision in which both eyes aim simultaneously at the same visual target, equally and accurately as a coordinated team. Since the two eyes are placed close together, overlapping of visual angles allows more accurate judgment of distance and of the object in front. Stereopsis or stereoscopic vision is vision in which two separate images of two eyes are successfully fused or overlapped into one image in the visual cortex of the brain. First, both eyes must have binocular vision. Then, because the two eyes are located in different positions, each takes a unique view from its own perspective, sending two separate images to the brain simultaneously for processing, from where the images are fused into one three-dimensional image. Rule number 1. When a person moves from bright sunlight into a dimly lit room, objects in the room cannot at first be seen, but they gradually become visible. In bright light, the circular muscles contract to narrow the pupil and reduce the overstimulation of the retinal cells by entry of light in the eye. In dim light, the radial muscles contract to dilate the pupil slowly to allow the entry of light, whose threshold at first is low to stimulate the rods for the object to be seen, but later improves to enable the vision as the pupil dilates fully. Part 2. In a dimly lit room, objects are only visible in black and white. The rods, which are sensitive to light of low intensity, do not respond to light of various wavelengths, causing the image to appear black and white. Roman number 3. Some nocturnal animals, like the cat, close their pupil to a vertical slit and squint in bright light. The retina of nocturnal animals is almost entirely composed of rods, with rhodopsin, which is particularly sensitive to low light level of light and break down so rapidly in bright light. The slit pupil and squinted eyes reduce the amount of light entering the eye to enable the rhodopsin form faster than it break down for vision to occur. Part 4. When trying to see a faint star in the sky, it's better to look slightly on one side rather than directly at it. When you look directly at an object, its image forms on the retina's fovea centralis, the yellow spot, which is packed with cone cells. Yet these are only activated by bright light, hence cannot see in dim light. Looking slightly away from the faint star moves the image off the fovea onto the part of the retina that have more rod cells, which are more light sensitive than the cones. If both eyes open and you press the side of one of your eyeballs, you experience a double vision. Pressing the eyeball distorts the eye position such that the two eyes are unable to look directly at an object in the same direction. Hence, the brain fails to blend together the images from the two eyes, causing double vision to be perceived. (35) Distinguish between exteroceptors and interoceptors. 2. Primary receptors and secondary receptors. Part B. Discuss interoception using suitable examples, highlighting the receptors, stimulus, location, structure, and the process of transduction. Part A. Distinguish between exteroceptors and interoceptors. Exteroceptors are receptors that are sense stimuli that arise in the external environment, while interoceptors, they are receptors that sense stimuli that arise from within the body. Distinguish between primary receptors and secondary receptors. Primary receptors consists of single neuron which is sensitive to a particular stimulus and transmit nerve impulses to another neuron or effector, while secondary receptors consists of a modified epithelial cell sensitive to a particular stimulus and a neuron. Discuss interoception using suitable examples, highlighting the receptors, stimuli, location, structure, and the process of transduction. The first interoception, we have the stimulus, which is temperature. The interoceptors are the thermoreceptors, which are the heat receptors and the cold receptors. Their location is in the skin and the hypothalamus. The structures, they have free nerve endings. And transduction, the temperature change, it opens or closes the ion channel in the membrane. Another interoception, the stimuli is touch. The interoceptors are the Meissner capsules and Merkel cells. Where are they located? In the surface of the skin. Their structures, they have nerve ending with elastic capsules. Transduction, rapid and extended change in pressure deforms the membrane. Another interoception, the stimulus is vibration. Interoceptors, they are the Panisian capsules. They are located deep within the skin. Their structure, they have nerve ending within elastic capsules. Transduction, severe change in the pressure deforms the membrane. Another interoception, the stimuli is pain. Interoceptor and nociceptors, where they located throughout the body. Their structure, they have free nerve endings. And transduction, use chemicals or changes in pressure or temperature. This opens or closes the ion channels in the membrane. Another interoception, the stimuli is the muscle stretch. Interoceptors, they are the stretch receptors. Where are they located? Within muscles. The structure is spindle nerve endings wrapped around the muscle spindle. Transduction, stretch of the spindle deform the membrane. Another interoceptor is stimuli is blood pressure. Interoceptors, they are the baroreceptors, where they are located at the arterial branches. The structure, they have got nerve ending over thin part of the arterial wall. Transduction, stretch of the arterial wall deforms the membrane. Thank you very much for participating in coordination and control. Questions 31 to 35. Please do like, share, and subscribe.